our guest speaker, who is an epitome of beauty and brain. She is an assistant professor of the Department of Biology in Ateneo de Manila, a registered microbiologist of the Philippine Academy for Biology. Our guest speaker graduated as magna cum laude of her class, the University of the Philippines, Los Baños, finished her master's degree at the same school with the degree of Master of Science in Molecular Biology and Biotechnology, and earned her PhD in Microbiology in University of Kansas. She embodies both grace and intellect. Some of her fellowship scholarship includes Fulbright Foreign Student Program, Internship Program for Asian Young Researchers in the field of Life Sciences and Bioengineering, Accelerated Science and Technology Human Resource Development Program, Panasonic Scholarship Program for Philippine colleges and universities. She is above and beyond all these achievements. She helps science world by publishing researches such as tobramycin adaptation enhances policing of social cheaters in Pseudomonas aeruginosa, bacterial quorum sensing and microbial community interactions, and the study on bioluminescent vibrio with antibacterial activity against the nosocomial pathogens Staphylococcus aureus and Klebsiella pneumonia. Friends, ladies and gentlemen, please join me in welcoming Dr. Rhea G. Abisado Duque. Let us give her a virtual round of applause.
Thank you, Ma'am Zara, for that very kind introduction. Let me just share my slide and we can get started. Alright, so hello everyone. So malahay na aga po sa kabos. So today I'll talk about antibiotic adaptation and cell-to-cell communication in Pseudomonas aeruginosa. So before I continue with this work, so let me take this opportunity to express my deepest gratitude to the organizers of this conference. So thank you so much for the wonderful opportunity to share our work with everyone here. Okay, so the work that I'm going to describe today is a part of a dissertation project that I did at the University of Kansas. So KU is a research university in the Midwest of the United States. So this was my home for 5 years, so home away from home. And I was able to pursue a PhD degree through the Fulbright program. So I was told that there will be many students attending today. So I thought of sharing my own journey in science, so to help those that are thinking of pursuing a career in science. So I am from Kasiguran, Sorsogon. So my interest in science, it started from elementary and high school days. Then I went to UP Los Baños for my undergrad. And so in this place, so I fell in love with the field of microbiology. So my very first work also involves research, and then I pursued a master's degree in the same school. And in this institution, so I was given lots of opportunities to improve my skills in research. So for example, I also went to Japan to do some research. And then after finishing my master's degree, so I joined the faculty of Ateneo de Manila University. So in that institution, so I learned how to become a teacher, an educator, a mentor. So back in UPLB, my favorite organism was the group called sulfate-reducing bacteria. So those are anaerobes. Then when I joined Ateneo de Manila, Manila University. So I did some collaborative research involving algae and the bioluminescent bacteria. Then in 2016, so I was given the opportunity to pursue PhD studies in the United States. And in KU, so I joined the Chandler Lab. So this laboratory is a quorum sensing laboratory. So it focuses on understanding how social behaviors evolve in bacteria. So if there are students here or young professionals thinking of pursuing graduate studies in the United States, in KU, or doing quorum setting research, so feel free to drop me an email. So I have my email address on the bottom of the slide. I'll be happy to answer any questions that you may have. Okay, so for today's talk, so I'll give first an overview on cooperation, quorum sensing, and social cheating in Pseudomonas aeruginosa. Then that will be followed by a discussion on antibiotic suppression of cheaters. Then lastly, I'll describe one example of effects of antibiotics beyond killing. So we'll talk about enhanced policing of quorum sensing defective social cheaters. Okay, so one of the most common collective behaviors in nature is cooperation. So organisms engage in cooperation to maximize the evolutionary fitness of individuals within a group. So here are some examples of cooperation in insects and other animals. So we observe cooperative building of nests in paper wasps, so food gathering in ants, and then in meerkats, so they use cooperative bullying, okay, to drive away potential predators. So cooperation reminds me of our bayanihan tradition. So I find this as a good analogy of cooperation at the society level. So in the world of bacteria, cooperation is commonly used as well, okay, to achieve specific goals. For example, bacteria engage in cooperation to build biofilms, to swarm, or even to produce virulence factors. So one of the model organisms for understanding cooperation in bacteria is the opportunistic pathogen Pseudomonas aeruginosa. So P. aeruginosa is also attracting lots of research interest nowadays because of its innate and acquired multi-drug resistance. So in North America, it's a major concern because it is a primary cause of mortality and morbidity in the genetic disease cystic fibrosis. It also has a role in nosocomial infection. So Dr. Rosana yesterday mentioned the SCAPE pathogens. So P. aeruginosa is one of those, and the SCAPE pathogens are the leading causes of hospital-acquired infections worldwide. And then just recently, so it has been shown that P. aeruginosa is one of the top co-infecting bacteria identified among COVID-19 patients. So cooperation in P. aeruginosa can be regulated by quorum sensing. So quorum sensing is a cell-to-cell communication system that is mediated by small signaling molecules. So in proteobacteria, so the small signaling molecules are in the form of acyl homoserine lactone or AHL. So quorum sensing is capable of regulating bacterial gene expression in a cell density dependent fashion. So for example, at low cell density, so the signals represented here by the yellow stars are still low in concentration. So under that condition, the bacterial cells behave independently from each other. So they display individual behavior. However, at high cell density, so the signals accumulate, okay? And when that happens, so group behaviors are switched on. So for example, in P. aeruginosa, so once quorum sensing is on, So they start to produce a blue-green pigment, okay, that can serve as well as a virulence factor. So in P. aeruginosa, quorum sensing systems are highly complex and they follow a hierarchy. So there are at least four known quorum sensing systems. So three are well studied. But for today, I'll just focus on the last RI system. So in this system, there are two regulatory components. So the last R, signal receptor, which acts as a transcriptional regulator, and the signal, so the AHLs. So in the last RI system, as AHLs are in the form of 3-oxo-C12. So these signals, they diffuse in and out of the cell. So synthesis of the signal is catalyzed by last I, and last I is also under the control of last R. So these signals are responsible for activating last R, and once activated, last R will induce the expression of the quorum sensing control genes. So last R is also considered as the master regulator. So if you inactivate last R, if you delete the gene, so that will result to a quorum sensing null mutant. So these quorum sensing null mutants are shown to be attenuated in acute infection models. So that illustrates the importance of last R for virulence. However, last R mutants are surprisingly common in infection environments. Okay, so I've said in the previous slide, last R is important for virulence, but then it tends to mutate over time. So we find a lot of last R mutants in infection environments. So last R is considered a mutation hotspot in P. aeruginosa clinical isolate. So it has been predicted that these last R mutants might have selective advantage in infection environments, or they could be acting as cheaters. There is also the possibility that both are happening. Okay, so I'll explain what cheaters are in a bit. So public goods production is a quorum sensing regulated cooperative behavior in P. aeruginosa. So a public good is an extracellular product which is expensive to produce for an individual cell, but it benefits all the members of the local group. So in this illustration here, so we have the cooperator in blue and then the public goods in yellow. So one example of a public good is the enzyme protease. Okay, so since it's excreted, so everyone can benefit from it. So even though only one cell produces it. So that's why it's considered as a very expensive bacterial product. So since public goods are costly to produce, so they are prone to social cheating. Okay, so cheaters, like the last Arnal mutants, they exploit cooperators, okay, by using a public good without paying for it. So they just utilize, for example, protease, but doesn't pay for a metabolic cost. So that's the advantage of a cheater. So a cheater, shown here in red, it can pop up in the population spontaneously. So this happens just spontaneously in a population of Pseudomonas aeruginosa. So since the cheaters may have a growth advantage over the cooperators because of their lower metabolic cost, so eventually they are capable of outnumbering, okay, the cooperators. So if that happens, that could result in a significant decrease in the public good production. So if the public good is required for growth, it becomes growth limiting, and it may come to a point in which the amount of the public good is not enough anymore, okay, to support the population. So if that happens, so the population may undergo collapse, it will crash, or in other words, the population will die. Okay? So in the context of population collapse, so cheating poses a risk. So how do bacteria maintain cooperation? So studies show that bacteria use cheating control mechanisms. So two of the mechanisms that I'll highlight today are policing and pleiotropy. So both of these are quorum sensing regulated. So in policing, so the cheaters are selectively killed by the cooperators through a secreted toxin. So examples of the toxins are hydrogen cyanide, rhamnolipid, and thiocyanine. So cheaters are selectively killed because quorum sensing doesn't only control the policing toxin, but it also controls the resistance factor. So since the resistance factor is only expressed in the cooperator, so the cheaters can be killed, but the cooperator can survive. On the other hand, in pleiotropy, so the co-regulation of public and private goods by quorum sensing restricts cheating. Okay, so the private good refers to the products that are required for growth and they benefit only the producing cells. So, for example, in P. aeruginosa, quorum sensing regulates protease, which is a public good, and at the same time, nucleoside hydrolase, which is a private good. So, nucleoside hydrolase is an enzyme needed for the catabolism of adenosine. So imagine a growth environment where the bacterium requires quorum sensing and adenosine is required for growth. So in that kind of environment, so the cheaters, okay, will be suppressed, okay, they will be restricted because it is only the cooperator that can produce the enzyme, okay, to catabolize adenosine, and at the it is the only type of cell that will benefit from that enzyme. So it doesn't share. Okay, so from the word itself, it's a private good. So it will not share the product to other types of cells. So the cheaters are therefore suppressed. Okay, so that brings me to the second part of my talk. So we'll now discuss antibiotic suppression of cheaters. So let me tell you a very short story that is another example of pleiotropy and became the foundation of our work on understanding what's the effect of antibiotic selection and adaptation on the evolution of quorum sensing. So I have here Chromobacterium, a soil bacterium that is really pretty. So it's purple. Okay. So this has an efflux pump, so a drag efflux pump, which is considered to be a private good. So that efflux pump is quorum sensing controlled as well. So in a growth environment where quorum sensing is required and then antibiotics are supplemented, so the cheaters are suppressed because they don't have the resistance factor. So they don't express the drag efflux pump. Okay, so they are also suppressed or controlled. So in P. aeruginosa planktonic cultures, so we also observe that quorum sensing contributes to antibiotic resistance. So here is a killing assay using the antibiotic tobramycin. So we're using tobramycin because it's a clinically relevant antibiotic being used to treat infections for cystic fibrosis patients. So on the y-axis, so we have cells per ml. So in the absence of tobramycin, so we did not see any growth difference between the wild type and the LAS-R. However, in the presence of tobramycin, so the LAS-R mutant is inhibited. So in P. aeruginosa, we don't know yet what is the resistance factor. So we don't know if it's similar to Chromobacterium. So to further test the idea that antibiotics can suppress cheaters in P. aeruginosa, so we use a laboratory evolution experiment. where we were able to monitor the emergence of last RNL cheaters. So what we did was we passaged P. aeruginosa daily in casein, okay, in the presence or absence of tobramycin for a month, okay. So to enumerate the last R cheaters, we patched the colonies on skim milk agar, and we identified them in terms of morphology. So last R mutants, they form flattened colonies with metallic shin. And they also have decreased proteolysis, although it's not obvious in these photos. So we're using casein here because casein, growth in casein requires expression of the quorum sensing controlled protease. So this is the plot of percent cheaters over time. So in the absence of tobramycin, so we observe the cheaters to emerge at around 7 days, and they invade the population eventually. And then at sub-MIC concentration, so 0.6 microgram per ml, cheater emergence was suppressed, so unlike in the absence of tobramycin. And then for stronger selection, so we subjected another group of P. aeruginosa populations to increasing concentration of tobramycin. So where we increased the antibiotic concentration by 50% every 4 days. So at that concentration, the cheaters were even more suppressed. Then about 5 years ago, so this is the time that I just started with my PhD studies. A paper came out predicting that cheating control would be lost or rewired during adaptation. So basically, so this paper here is saying that with antibiotic selection, bacteria can accumulate genetic mutations. So genetic mutations that can increase antibiotic resistance. And in some cases, so these mutations can have secondary effects on other traits. So for example, cooperation. So with that prediction and the observation that last R mutants are abundant in infection, so we became interested on understanding the impact of adaptation, so antibiotic adaptation, on the dynamics of cooperation and cheating. So we started our investigation by isolating bacteria with functional quorum sensing system from each of the populations at the end of the evolution experiment that we carried out. So we got 3 isolates from here, so no tobramycin evolution experiment, and we also got 3 isolates from here and from this condition. So for the interest of time, I'll skip the nitty-gritty part of characterizing the antibiotic adapted variants. But in summary, so we found that adaptive mutations accumulate in the tobramycin evolved variants. So 2 of the most common mutations are in the PTSP and FUSE-A1 gene. So all the tobramycin evolved isolates that we have mutations in PTSP, a gene coding for a phosphotransferase enzyme. And the next commonly mutated gene is FUSE-A1, coding for an elongation factor. So both of these genes are not mutated in the ancestral wild type, as well as the isolates from no antibiotic evolution experiments. This suggests that these mutations are adaptive mutations in response to tobramycin. So that brings me to the last section of my talk today. So this is one example, okay, as I've said, one example of an effect of antibiotic adaptation on the evolution of quorum sensing. So let me tell you more about PTSP. So PTSP codes for the first enzyme in the nitrogen phosphotransferase system found in gram-negative bacteria. So the system is illustrated here. So the product of PTSP is responsible for the phosphate transfer from the phosphoenol pyruvate, okay, and the second enzyme. So in E. coli, the nitrogen phosphotransferase system regulates diverse physiological changes in response to nitrogen starvation. So in simple terms, so this system, the nitrogen phosphotransferase system, helps E. coli adapt, okay, in nitrogen starvation. So it changes physiology of the bacteria so that it will survive. In P. aeruginosa, it is not clear if it has the same function. However, it is known that PTSP mutation is a common genetic adaptation to tobramycin. So our interest on PTSP, it started from a previous report that PTSP inactivation increases LASR activity. So we measured the LASR activity in the tobramycin adapted variants. Okay, we used a GFP reporter fused to a LAS-I promoter. So as illustrated here, LAS-I is responsive to LAS-R. So whatever activity we detect is a measure of the LAS-R activity. So here is the relative LAS-I GFP of the different
Recording in progress.
in aeruginosa and measured the LAS-R activity. So we also observe about twofold difference in the activity of the PTSP mutant and the wild type. So very similar to the tobramycin adapted strains. So since PTSP increases LASR activity, so we predicted that that may increase the metabolic burden associated with keeping a functional size, that inactivation of PTSP would actually result to an increase in susceptibility to cheating. So to test that hypothesis, we passage the tobramycin adapted variants with PTSP mutations um, daily in casein, and this time we did not add tobramycin. So to promote cheating, okay, so we did the evolution experiment again for a month, and we monitored or detected last RNL cheaters, so using morphological features. So here is the percent, the plot of the percent cheaters over time. So just like what we've seen in the previous slide, so the last are now cheaters in for the wild type. So they pop up very early and then increase in number. However, the cheaters, okay, on or coming from the evolved isolate, so the tobramycin adapted populations, the cheaters were suppressed. And this is surprisingly the opposite of our hypothesis. Okay, so we did not even observe cheaters, okay, in the evolved population too, okay, for at least 32 days that ran the, the experiment. So apparently we were wrong. So our prediction was completely wrong. So we also tested the effect of PTSP in activation. So we made a clean deletion mutant of PTSP. So to make the comparison more straight, days it take them the number of days it take for the cheaters to emerge and consistently increase in number. So the wild type, okay, here is showing an early cheating phenotype. Okay, we included a strain with a CTX empty vector, so as a control, because in the complementation experiment that we did, so we used the CTX system, okay, to complement or to put back, okay, the wild type PTSP gene. So it is just showing the same result as the wild type. So that means that the CTX system is working. So for the PTSP mutant, so same with what we have observed for the tobramycin evolved isolates, we also observed delayed cheating, okay, relative to the wild type. And we can restore the cheating phenotype of the wild type, okay, in a PTSP mutant by introducing the wild type PTSP gene on the neutral at P site, okay, in the genome. So that confirms our results that that the PTSP mutation is actually delaying cheating, so instead of improving cheating. So how does PTSP inactivation decrease cheating? So from all the information available to us, so we thought that PTSP inactivation might decrease cheating through policing. So let me remind you how policing works. So the cheaters here are selectively killed by quorum sensing controlled toxin produced by the cooperator. So selective killing occurs because the toxin resistance factor is also quorum sensing controlled. So the cooperator is protected. So we were thinking that PTSP inactivation can actually enhance policing, okay, or can enhance production of toxin, which in turn can enhance policing of cheaters. So the first question that came to us is, does the PTSP mutant produce a toxin? So the answer is yes. Inactivation of PTSP is known to enhance thiocyanine production. So thiocyanine is a phenacin, and it is responsible for the blue-green color of P. aeruginosa cultures. So here we could see the difference in the intensity in the pigment produced by the wild type and then the PTSP mutant, so indicating that there is more thiocyanine production in a PTSP mutant. So thiocyanine synthesis involves several proteins as illustrated here. So the products of these two operons here convert choresmate to PCA. So PCA is phenacin 1 carboxylic acid. And then PCA is converted to 5-methyl PCA through PSM and 5-methyl PCA into thiocyanine through FEB. So PCA can be converted as well to another type of phenacin, phenacin 1 carboxamide through this H. So all these phenacins and thiocyanine intermediate, so they were reported to be toxic. So we tested each one of them. So we deleted the genes to create the mutants of the different phenacin and thiocyanine intermediates. Then we collected the culture fluid of the mutants. So the filtrate, we use it as a culture medium for a PTSP last R double mutant. So we're using this strain as the cheater. And we quantify the CFU per ml of the cheater upon exposure to the filtrate. So we have here the CFU per ml of the cheater, okay, how they grow. So this graph will show you how the cheater would grow once exposed to the filtrate. So we have seen that upon exposure to the filtrate of the wild type, so its growth is not inhibited. However, exposure to a filtrate of a PTSP mutant results to growth inhibition. deletion of the two operons, okay, in the PTSP background, okay, eliminates the growth inhibitory effect displayed by the PTSP mutant. We did not also observe growth inhibition when we deleted FIS-M and as well as FIS-S, while we still observed growth inhibition with a FIS-H mutant. So these results here suggest that the policing toxin is thiocyanine after all. So to further confirm our results, so we tested commercially available thiocyanine. Okay, so we used a PTSP FIS-S filtrate. So from the previous slide, we have seen that this doesn't inhibit, okay, the effect, the growth of the cheater, okay, the PTSP class R cheater. And another condition that we included in the experiment is a filtrate coming from the same strain, and then we supplemented it with 10 microgram per ml of thiocyanine. So here we have the CFU per ml, okay, of the strains exposed to these filtrates. Okay, so we have the wild type and the cheater. So the double mutant PTSP and LAS R. So exposure to the PTSP FIS-S filtrate, we did not observe any growth inhibition. So the wild type is just similar to that of cheater. However, exposure to commercially available thiocyanine, so that resulted to growth inhibition in both strains. However, we observed that the thiocyanine is significantly active against the cheater. So to wrap it up, PTSP mutations are a common genetic adaptation to tobramycin. And inactivation of this gene increases LASR activity and as well as thiocyanine production. Thiocyanine generates toxic hydroxy radicals, okay, which could kill the cheaters. So the cooperator is somehow protected by producing enzymes that can neutralize the free radicals. So examples of those enzymes are catalase and superoxide dismutase. So since these enzymes are quorum sensing controlled as well, so the last are cheaters. So the cheaters are more sensitive to thiocyanine killing, okay, relative to the cooperator. So overall, so tobramycin adaptation indirectly contributes to the stability of cooperation by selecting for a mutation, like a PTSP mutation, that could enhance policing of cheaters. So hopefully at this point, you are convinced, okay, that quorum sensing in the aeruginosa contributes, okay, to tobramycin resistance, to antibiotic resistance. And moreover, so we have seen examples on how antibiotics affect the cells. Okay, so antibiotic effects, effects of antibiotics beyond killing. So antibiotics don't just kill bacteria, so they can also modify the physiology of the bacteria. So in the context of quorum sensing, so antibiotics could alter the quorum sensing activity, and it can also alter the dynamics of social cheating. So currently, we are also exploring other effects of antibiotic adaptation on quorum sensing. So I already left the lab of Dr. Chandler, but we still continue to collaborate with each other. So this is another good output or good result of my PhD experience in KU. So one of the things that we're doing is to understand, okay, the mechanism of population collapse. So another phenomenon that we have observed with evolved isolates, so some of them acquired genetic mutations that induces population collapse. So I'm showing you here the percent cheaters that pop up in an evolution experiment for a wild type and a gene with a mutation. I mean, a strain with a mutation on a gene MTPA. So for the wild type, so cheaters pop up very early and then increase in number, but the population remain stable. So if you will look at the cultures, so it will look like this, very happy. Okay, so it's blue-green, so that indicates that the cells are healthy and they continue to grow and proliferate. However, for a genetic mutant for MTPA, we observe that at some point it will suddenly collapse. So from this blue-green culture here, it will become like this. So indicating death, okay, collapse or crashing of the population. So we're trying to understand what's the implication of this and how does it happen. So MDPA here is involved in dipeptide metabolism. So at this point, so we're trying to find the mechanism behind it, and we observe the same phenomenon in other genetic mutants as well. So NTPA is just one of them, and we think that they have different types of mechanisms. Then another project that we're also working on is the understanding of the mechanism of the altered role of LASR in antibiotic resistance. So earlier, I have emphasized that quorum sensing contributes to tobramycin resistance in P. aeruginosa. However, our evolution experiments also provided us with tobramycin adapted strains with reverse phenotype, with altered phenotype. So we have here the tobramycin MIC on y-axis and then for these strains. So for the wild type, the ancestral wild type, we usually observe a higher resistance, higher MIC for the wild type relative to the last R mutant. So it's not very obvious here, but if you will look closely at the figures, so Lazar mutant has lower MIC compared to the wild type. So the difference is not very significant. Um, However, with it is known that once genetic mutations that induce resistance accumulate, so even if it's very small, that becomes very important in the clinical setting, and that also dictates the survival of the organism. So even if it's very small, we consider it as a very important factor in terms of antibiotic resistance. However, in an evolved isolate, we call it T1. So we observe that it is the opposite of what we observed in the ancestral wild type. So whenever you delete last R, it becomes even more resistant, which doesn't make sense at all. So we can we know that the phenotype is real because we can phenocopy it in a delta last I mutant. So we deleted last I from T1 and we observed the same phenotype. We can restore the T1 MIC level by supplying external source of AHL. So at the moment, so we're piecing apart what's the mechanism behind this. So what makes this resistant relative to the strain with a functional quorum sensing system, with the goal of understanding what's the change that occurred with quorum sensing. So this is another trajectory by which quorum sensing would evolve once exposed to antibiotics. And the last project that we're also working today together with my students in Ateneo de Manila University. So we're also screening for natural compounds that can serve as quorum quenchers. So quorum sensing inhibitors are promising alternative therapeutics. They can potentiate the effects of antibiotics. So we're looking for screening for natural compounds that can do that function. So we know that quorum sensing is important for virulence and pathogens. So and P. aeruginosa is a multi-drug resistant pathogen. So it would be good to search for alternative therapeutics. So with that, so I would like to acknowledge my amazing mentor, Dr. Chandler, and then all the members of the Chandler Laboratory. So this is us before the pandemic. And then at the height of the pandemic in Kansas, so we had a lockdown, but we still do science over Zoom. Okay, and this is us before vaccination, and then after everyone is fully vaccinated, so we were already brave enough to be close with each other to take a photo, and then the funding agency supporting our project, my Fulbright Fellowship, and as well as my collaborators or our collaborators from the University of Washington, Dr. Dandikar and Nicole. And I would also like to give a shout out for Team Pseudomonas of Chandler Lab. So in the Chandler Lab, so we're working on 3 different kinds of organisms that can perform quorum sensing. So one is on Pseudomonas, another group is in Chromobacterium, and then the third group is Burkholderia. So we study how quorum sensing evolved and its importance in pathogenesis and virulence by using those 3 different types of bacteria. So these are the members of the Team Pseudomonas group. And since I'm the third oldest in terms of seniority in the Chandler Lab, so I had the chance to work with many of the students in the lab. So some of them are PhD candidates and a lot of amazing undergrads. Okay, so I find this as a very important experience, okay, in my PhD training in the Chandler Lab, because it gave me the opportunity to hone my mentoring skills. Okay, so um, that's the end, okay, of my presentation. So thank you so much, everyone, for your attention. Um, it, I am happy to answer any questions that you may have. To God be the glory.
It was truly a comprehensive and informative talk, Ma'am Rhea. Thank you so much. Let us give Ma'am Rhea a big round of applause, please. Okay. Thank you so much, Ma'am. We learned a lot from this talk on the cooperation, quorum sensing, and social cheating in Pseudomonas aeruginosa, the suppression of these cheaters, okay, and of course the controls of the cooperating populations. So now we would like to open the room or the chat box for your questions or clarifications to Dr. Rhea. You may now input or chat your queries in our Zoom chat box. Okay? We actually allotted the 30-minute period for Q&A. So, here, I received a direct message, ma'am. We have here a question, Ma'am Rhea. What are other applications of quorum sensing?
Yeah. So in many bacteria have different types of quorum sensing system. So earlier I mentioned bioluminescent bacteria. So that's another form of quorum. Of phenotype that is regulated by quorum sensing. In P. aeruginosa, at least 10% of the genome of the organism is regulated by quorum sensing. So you can imagine how broad, its effect on the physiology of the organism. But so In my PhD lab, so the main goal is to understand how this quorum sensing evolved, and so that we can address concerns related um, to virulence and pathogenesis. So since quorum sensing can regulate virulence factors, so you can explore it, you can exploit the system on how to control the organism. So many microorganisms now are becoming multi-drug resistant. And so there's actually a need to find alternative therapeutics. So by understanding quorum sensing itself, um, there's a big chance that we can find ways on how to control control the organism or how to treat them. Um, so in quorum sensing as well, so for other types of bacteria, so there are different types of functions regulated by quorum sensing. So it's a long list probably if we would be citing each one. But it's more on regulation. So sometimes what works with controlling the organism, it's good to target the regulatory factor rather than the gene itself, because by doing that, then we can target lots of genes instead of just doing one resistance factor, for example. And then in P. regionosa, quorum sensing also controls lots of byproducts. Okay, so for example, I mentioned thiocyanine, there's rhamnolipids, so all of those are controlled. And some of these byproducts are used in industries. So if you want to produce something out of the originosa, you can manipulate quorum sensing system. And then biofilms as well. So that's under, quorum sensing has a role or playing a role in controlling biofilms.
Thank you, Ma'am Rhea. I hope that answers your question. Ayaw niyang magpakilala, Ma'am. Francis from from Dr. Francis Alegado. Thanks, Doc Rhea, for your very informative talk. Thus, the quorum sensing mechanism provides better mechanism against antibiotic resistance compared to other mechanisms of antibiotic actions.
Thank you, Dr. Alegado. Hi po. Um, yeah. So, with current targets of antibiotics, so there is this. The main problem is that bacteria can develop resistance against those agents. So, what is what has been promising with quorum sensing agents? So far, what is known, so there is lesser tendency that bacteria can become resistant to antibiotics. But first, I would say that there's still a lot of unknowns. But so far, we have that premise that with controlling quorum sensing against multi-drug resistant bacteria, so there is lesser tendency that they will become resistant in the future. And another way of applying quorum sensing quenchers is you can actually use it in tandem with existing antibiotics. So there are studies showing that it can potentiate the effects of antibiotics. So if an antibiotic doesn't work anymore against, for example, Pseudomonas aeruginosa, you can combine it with a quorum quencher or a quorum sensing inhibitor, and it will work again. Thank you for your question.
All right. Thank you, Ma'am Rhea. We have here another question from a regular member of PSM, from Sir Ivan Joshua Castor. Are the cheaters still considered to be as pathogenic as the normal bacteria? And were there cases where the cheaters performed quorum sensing with other cheaters? Thank you po.
Yeah, that's an interesting question. So the first question, do last R mutants or do cheaters still considered pathogenic? So in laboratory experiments, so they have seen that they are attenuated, so less or not anymore pathogenic when they are in, allowed to infect a host system. But actually, at the moment, so lots of developments have been acquired out of Pseudomonas research. So they have discovered that the different quorum sensing systems in Pseudomonas aerogenes are highly complex. So if you mutate one of them, it will find a way so that the other systems would compensate and will make them do their function again. So for example, they found strains with mutations in BLAST-R that can still become pathogenic because the other system makes it pathogenic. So for example, one system that is discovered is the real system. So we have LAS-R, we have real system, and then we have the PQS system. So those are the three well-known quorum sensing systems in P. aeruginosa. So there are strains where you delete last R, you make it inactive, but then the real system would work again. So in terms of quorum sensing inhibitors, so that's an important consideration that you have to think about. So your quorum sensing inhibitor should be good for all of them. And then the second question was if can you remind me, Ma'am Syra? So the cheaters.
Were there cases where the cheaters performed quorum sensing with other cheaters? That's it po, ma'am.
Okay. Yeah, so in observation, so laser mutants or laser cheaters, so they don't perform quorum sensing system at all. So if you measure the different parameters that indicate quorum sensing system is off, the only exception is the first example that I mentioned. So, but it only happens if there's additional mutation, okay, that is present in the bacteria. So, for example, you have a last R mutant, but with a functional REL system. But then, if this organism has a mutation, for example, in MEX-P, okay, which is a part of an efflux, so they have observed that the organism can become or can perform quorum sensing, certain quorum sensing functions. So that's the difficult, complex part of dealing with quorum sensing systems, especially in Pseudomonas. So it's very highly complex. So I guess the short answer is yes.
Okay, that's it. Ma'am Rhea, we have a question from Ma'am Kathleen Pahoyo.